But what's amazing about these tools is that they've gotten so good that I was talking to PhD students and postdocs there who had been in the lab before the deep learning revolution, been in the lab during and after the deep learning revolution, and they're like, everything's changed. Proteins are abundant in our body, doing lots of different and amazing things to keep us alive. They are a big part of medicines too, like synthetic insulin, which we talked about last episode, antibodies, and protein vaccines, just to name a few. And over the last decade, it's become possible to use AI to predict the structure of proteins much more accurately and use that knowledge to design new proteins that have never been seen before in nature, improve existing proteins to use for medicines and other industries and agriculture and more.
Welcome to Hard Drugs. I'm Saloni Datani, and this is Jacob Trefethen. I'm a co-founder at Works in Progress magazine, and Jacob leads science and global health R&D funding at Open Philanthropy. And in today's episodes, we're talking about proteins, how to improve proteins, and how to design new proteins.
Last episode, you taught me about insulin, a protein that I know is pretty useful and the body makes naturally. And that's also useful if you make it outside of the body and provide it as a medicine for people who need it.
Right. It's used for diabetes. Insulin helps control blood sugar levels. And yeah, so in the episode we just did, we talked about how it used to be extracted from animals in the early 20th century. And then in the 1970s, people developed a way to reproduce it in bacteria that were used in this giant bacterial churning soup machine. And in such a way that you could scale up this protein much more than you could with, animals that are being factory farmed, so that they can be used for many more diabetes patients around the world.
And that was a game changer over the last 50 years. Today, we want to speed through to the last 5 years, really, and talk about some game changers of new AI technologies that help you not just take a protein nature has designed and reproduce it, but actually tweak or improve a protein that nature has designed for an even more useful medical purpose. And that really started getting more and more possible with tools invented since 2020. So,
Whoa, that's very recent.
It's very recent. And, people probably have heard of AlphaFold, which AlphaFold 2 came out in or was first used in 2020.
Mm-hmm.
And some of the other tools that help you improve on existing proteins were made since then, 2022 and since. So,
but first,
yes,
why proteins? Why are proteins so cool? We talked about it a little bit in a previous episode. I think there are a few things. One is they have like a single protein could be doing lots of different things. You can have like modular parts of proteins that are doing a bunch of things together. Like maybe one part is doing an enzymatic reaction, another one is binding to something that tells it how to speed up that reaction or to slow it down. Maybe there's some other like signaling parts that are like, hey, protein, stop working now, and stuff like that. So that's like one. I think the other is, yeah, like it reacts to the environments, like changes in temperature or acidity or things like that could change how much the protein is doing something. Also, they're quite small. Like they're like if you were a chemist who was trying to make some reactions happen and you're doing like a series of reactions in a bunch of machines, a protein can do that at a like tiny scale. Like this tiny protein is doing all of that stuff. And it's like super specific as well, right? Like it could bind to a tiny molecule or a metal or another protein or another receptor or like so many different things. And it's like super specific. So lots of reasons.
Lots of reasons. And I want to convince you now that that's not enough
Oh,
In that,
I wasn't expecting that.
nature has not evolved all of the proteins we might find useful in our body.
Mhm.
And that's why these new AI tools are pretty useful.
Okay, so the potential uses of proteins are very, very large, I think.
Yes.
But the ones that we see in nature are not necessarily. And maybe they've only evolved to do certain things in certain environments. Is that right?
Yep, exactly. And I want to make it specific by talking about, like we had insulin last episode.
Mhm.
Let's talk about a medicine that we don't yet have that we might be able to make with the help of new tools. So
What's the protein?
I picked one called CypA.
CypA.
This is a protein which is on the outside of a bacteria. The
Mhm.
bacteria is Strep A. And I, as some of my friends well aware, am a bit.
You're a fanboy.
I'm a fanboy, or
Well,
really,
you're
I
a
hate.
hater.
I'm a
Yeah.
hater of Strep A. And the reason is that Strep A, as a bacteria, kills 500,000 or maybe more than 500,000 people a year because it can lead to different diseases if you get repeated infections. So in particular, it leads to rheumatic heart disease, which used to kill a lot of people in the US and the UK and other high-income countries, still kills a lot of people in lower and middle-income countries. And I mean, as a side note, Strep A is also the bacteria that, you ever seen that flesh-eating stuff that goes up your leg and then eats your whole body?
Ooh. Well, I guess I've seen a few different diseases that do that, but yeah, that's scary.
Yeah, Strep A is one of the biggies.
And wait, is that also strep throat?
Yes, strep throat and scarlet fever and different names for these diseases. Yeah.
And so I think the one thing that I know is about this is like the bacteria maybe first causes an infection of the throat. So it first causes scarlet fever or strep throat. And then maybe later on people develop heart disease.
Yes,
from it.
exactly. Because what happens is your body's making an immune response to that bacteria that it can actually get confused and start attacking your own heart valves,
Oh.
which not so good.
That's terrible. And yeah, you also wouldn't usually, I think the classical way people think about infections is not that they're linked to heart disease, but in fact, there are
Oh, yeah.
many ways that they can be connected.
Absolutely. So we don't have a vaccine against this. It would be pretty nice if we had one. And the way that you make vaccines often in a modern context is you try and take protein antigens from a given pathogen.
What is an antigen?
An antigen is a part of, in this case, this bacteria that prompts a immune response in your body. So there's many different things that are involved in Strep A, the bacteria, reproducing and living. But some of them are better things to target for your antibodies and other parts of your immune system.
Right. So if I was trying to recognize you
Yes.
from far away, it would be more helpful for me to know what your face looks like.
Yes. Yeah.
And what your clothes look like, because you might change your clothes.
That's exactly right. And sometimes if I'm really stealthy, I might change my face, but that is a bit more costly.
Ooh. You know, I used to watch these. Well, I didn't watch them, but on Indian television, there were soap operas where that would be ongoing for like years or decades. And sometimes the actors would not be interested in working on that show anymore. And they'd like leave the show, and the producers would replace the actor with someone else.
That's what we call a stealth pathogen.
And so they would have to explain what happened. And usually it would be like they had cosmetic surgery or like they got into an accident. So they write off the old actor from the show, but they still want the character in the show. So they just get someone else to play them. And they're like, oh, now they look different.
I'm always impressed that the main characters in Harry Potter were still the main actors by the end, because that was a decade of anyway. So
yeah, that's impressive.
Well done. So I'm Emma Watson.
You're Emma Watson?
No, I'm not. I'm actually Jacob. But I am Strep A. That's what I am.
Okay. Oh. Okay.
And we are trying to find antigens on the outside of Strep A that could be used as a vaccine or as part of a vaccine. And there's a lot of attempts to do this already. So you might not end up needing AI, but AI is starting to help. And I'll walk you through an experience I had of sort of playing around with these tools to get to grips with them a bit more. So I went to visit the University of Washington in March this year and got to spend a couple of weeks at the Institute for Protein Design, which focuses on a lot of protein design tools and has invented some themselves. And while I was there, I focused on this one antigen, CYP-A, and tried to use a tool called Protein MPNN to revise it to make an even better antigen or immunogen.
Okay. So let's say, again, I am trying to recognize you from far away.
Yes.
And I know what your face looks like.
Yes.
Why would I want to improve that?
Okay, so let's start with what CYP-A looks like. So this is a protein that's stuck on the outside of the bacteria. Now, that's a good start because your antibodies can access what's on the outside of a bacteria much more than they can access what's on the inside.
Right.
So that's a good start.
They don't fit in. They can't get in.
Right. They don't get in. Now, you might say, okay, great. Why don't I just take that thing and use it as a vaccine? Well, you could try that, and people do try that. You can make changes, though, that improve on the properties of that protein from the point of view of making an actual vaccine, making a product that you could inject. So, for example, any given protein, it's not guaranteed to be that easy or cheap to make. So you can make it in different systems, different bacteria and yeast. And if you have to make a protein in mammalian cells, for example, instead of in yeast or in bacteria, that's going to be more expensive. So that's not great from a product development point of view. It also might not be as stable as it is in its natural occurrence on the outside of a bacteria. If you're plucking it out of this membrane and you're just putting it in, well, are you sure that's going to not just clump up together with other versions of itself if you got a lot of them? You sure it's going to be soluble in water, which is a pretty requirement property for a vaccine? And are you sure it's going to not kind of deform into something once it's not plugged in the same way? And so you want to make alterations to that too. And then finally, are you sure if on its own, it's going to be as immunogenic as when it was in its natural form on the outside? Immunogenic in the sense of prompting the immune response we're looking for.
Right.
And this particular antigen, it's about 1,000 amino acids long.
Okay, that's like moderate, I guess.
Yeah, it's sort of medium big.
Yeah, we talked about some tiny proteins before, and they were like 20 to 30 amino acids. And then we talked about a huge protein, Titin, which is like 33,000 something amino acids.
Yeah, so this one's sort of in between those. And if you can get away with it, it might be nicer to take only the bits that really matter for the immune response and make it smaller. So if you can still prompt an immune response, it'd be kind of nice if it was only 200 amino acids long. Yeah. It might be cheaper to produce, for example. It might be if you put it on a scaffold of think of a soccer ball, and you want to put lots of different antigens on that soccer ball to prompt an immune response. If it's smaller, you might be able to fit more on.
So I'm unfortunately still thinking about the analogy where I'm trying to recognize your face, and now I'm like, the soccer ball has little Jacob faces all over it.
Well, you definitely recognize that.
It would be scary, though. But also, this makes me think of like, okay, in history, we didn't have vaccines that were just one antigen. They weren't just one part of the thing. It was usually like the entire virus or the entire bacterium, and that would be killed in some way. Maybe it's like chemically inactivated, or it's like attenuated, so it's like put into cell culture and made to evolve into something that doesn't infect us or cause harm.
Yeah.
And then we're using the whole pathogen. And this is very different. This is like a very precise part of the pathogen.
Yep.
And it turns out sometimes we only need that to recognize the whole pathogen. And maybe that's also useful because there are other parts of the pathogen that are harmful to us in some ways.
Exactly. And in this case with strep A, if you know that the whole pathogen prompts a immune response that you might hurt your own heart, then you sure enough want to get rid of some of it.
Right.
Yeah.
And then are there other reasons that we would want that? Like what is this? What is the bacteria doing? What are the proteins doing?
Well, actually, in this case, yes. So I'm going to take a quick detour that is not central to the point. Are you okay to bear with me?
Yes.
Okay,
yes.
great. So this particular antigen is kind of messed up.
Oh.
So what it does is it hangs out on the outside of the bacteria, and it's a peptidase. So imagine it kind of looks, for people watching the video, it's
like a crocodile.
It's like a crocodile. And what happens is that it's evolved to cleave or chop in half, say, some signaling proteins that our body sends to the pathogen to actually recruit even more immune cells. So C5a. So if you send those immune proteins to it and they're going to bring some buddies, it's going, nope.
Oh my God.
Nope.
So wait, so this bacterium is trying to infect me, and then my immune response is trying to attack it in response.
Yes.
And it's sent out all of these immune cells to attack it.
Yep.
The signals just get cut up.
They're just getting cut off. So
that's really sad.
It's so sad because, this is one of those things where like our immune system is pretty good and it is getting activated and it's going to try and mess up that bacteria. And the bacteria is like, no, you don't.
Oh, that's scary. And so if we could somehow change this this protein or this bacterium, we could find a way for us to recognize it without it cutting up those signaling proteins.
Correct. Now, the reason this is a detour on the main point is that is not that hard. So you can actually just look at this cleaver in question and imagine, it's the crocodile jaws. You just pick the amino acid residue that's most at the jaw. I'm speaking loosely.
So you put something in. So you're saying you could block the cutting by putting something into it.
Yeah, essentially. But I mean, just changing the string of amino acids so that at the position that's usually histidine 193, you put leucine, or I forget what, you put a different amino acid. You actually only have to make one change to this 1,000 amino acid long protein, and it will no longer perform the harmful function.
That's amazing.
That is, you don't need AI for that.
Right.
That, you can validate that you've neutralized that aspect of it.
Okay.
And so just stepping back, we can get to a pretty useful initial test of a vaccine without AI, where what we did was we said, we got this bacteria that's invading us and we don't like. We took one of the things on the outside that antibodies do bind to, and we're just going to inject that after changing a residue so it doesn't hurt us.
Right.
And what will happen if that works well? The immune system broadly, but let's just visualize antibodies, will bind that, and you'll generate a adaptive immune response that remembers how to produce those antibodies. If you get an infection of strep A later, those antibodies are going to come and hit that bit of it.
So later, basically, I recognize your face. And if you were in a different place, like in a sand pit on a beach, you've dug up a hole.
Who put me there?
I don't know. Maybe you did it yourself.
Fair.
Don't people like doing that? And then, so maybe you're inside and only your head is sticking out. And I'm like, that's Jacob. I know that it's you. And I don't need to see the whole body to know that.
Yes. And actually, in the context of the immune system, it's a good metaphor because you're trying to neutralize me. So you probably brought a, bow and arrow.
Oh.
And if you shoot me in the head, that will actually kill all of me.
Oh my.
I'm neutralized.
Right.
So you don't have to shoot my hands. You don't have to shoot my legs. You got that kill shot in the head.
Right.
So well done. While I was in the sand, but I couldn't.
Well, yeah, you probably couldn't move at that point.
Move. That wasn't my fault, but.
Yeah.
So. AI.
Wait. This protein, we just have the one protein. I think the other thing that's really helpful about this is that it's not the entire bacteria that's like harming us. It's not like invading different parts of our body and things like that. We've just honed in on your face. And so that's really useful. And that's like, okay, we can recognize this when it comes later on. And then maybe if there are other types of, if you had an alter ego who shared your face, or if there was a different species, or there was a different, strain
Yes.
that had some similarities
Different
like
strain
your
of Jacob.
face, but
Yeah.
different body,
Right.
then I would still be able to recognize that as
Right.
well.
You could neutralize jacked Jacob. You could neutralize short Jacob. Yeah. Absolutely.
Cool. Okay, so we talked about protein vaccines, and we can improve them.
Yes.
This is not good enough.
Yeah, so I mean, it may be. I think in this particular case, it probably wouldn't be. And you might want to improve that head of mine. Maybe give me a little bit of Botox or something, so that I'm even more recognizable to
Right.
your recognition system. So there's this new tool, Protein MPNN. Protein MPNN was made in the Baker Lab, David Baker, who just won the Nobel Prize alongside the AlphaFold inventors, by him and by some students. In particular, Justus de Porras, I think, was one of the lead authors, which basically takes the structure of a protein and predicts what sequence of amino acids leads to that structure.
Ooh.
So structure in the sense of, think kind of visually 3D, where do the residues, where do the carbon atoms in this backbone actually appear once the protein's all folded up? And sequence, think, what is the string of amino acids?
So I'm imagining a bead of strings, which is the protein. And this bead of strings is like folded up into this larger structure. Maybe it's like a knitted ball that like a kitten plays with or something. And it's like it's not as symmetrical or anything. And we are trying to predict exactly what amino acid is each of the beads. And we know what the string of beads looks like, what that shape of the folding is. But we're trying to predict each of the beads.
Correct. Exactly right. And we may not have there may be multiple strings that end up folding up to kind of similar. So you're not always just trying to predict, what's the exact string. You're often trying to give me some hypotheses here that you then want to test. So in the case of this antigen we've been talking about, you can say, I want to make this smaller. It's currently made of 5 domains, which are these subunits of the protein that self-assemble and then assemble together. Could I generate the same immune response with just 2 of them, the 2 most important ones? So let's just chop off. Let's just chop off the other 3 domains and look at these 2 together. And then I'm going to ask Protein MPNN. I'm going to say, and I did this when I was in University of Washington. I was like, okay, here's what it looks like. Here's what I want it to look like.
How do I make that?
How do I make that? Once you chop off some of those domains, you can't immediately use what's left, because there will be amino acid residues that have evolved to be in particular points inside the protein that are doing great. But if they're suddenly exposed, some of those may be hydrophobic. Hydrophobic, for example, which means that what you've just created is not going to be soluble. So you're going to want to mutate the amino acid sequence a little bit here and there so that you're not exposing, for example, hydrophobic residues.
So I remember learning a little bit about protein structure, and one thing that I remember is, so if a protein is soluble, then the outside has to be attracted to water, I guess, hydrophilic. But the inside is usually hydrophobic.
Right.
And that makes it like oil, say. So if it was hydrophobic on the surface, then it wouldn't.
Yep.
Yeah, it wouldn't dissolve.
Yep. But we want it to.
We want it to dissolve.
So we're going to have to make some changes.
Because I guess, well, if I'm thinking about the environments of a protein, it's usually Maybe it's in the blood or it's in cells. It's in these places where there's a lot of water content.
Yeah. I mean, most of life's chemistry happens in aqueous solution. So we got to be ready for that.
Right. And so that would be helpful, not just to cut it down so you could only make 2 domains, but just generally, like, let's say you just had a mystery protein in front of you and you're thinking, how do I recreate this?
Yeah.
I would maybe imagine, I don't know if people actually do this, but like I would just imagine, I don't know, I'm a pharma company or I'm a biotech company and I found a protein and it's doing something really cool, or I found my competitor's protein. And I'm like, how did they make this? I want to make this.
Cool. Yeah.
And then I would use this tool to try to figure out the letters that make it. And you know what? I really like the name protein MPNN because I'm thinking it's trying to predict each of the amino acids in the bead of strings. And you represent each of the amino acids with a letter, right? And so it's like MPNN.
Oh, nice.
Does that stand for an amino acid chain? Maybe it does.
I wish. I'm going to look it up. It does!
Oh my God, it does. Wow, what does it stand for?
It stands for methionine, proline, asparagine, and asparagine.
Whoa, that's very smart. So yeah, I mean that's a really good way to think about it, because it's okay, you've got the structure. Now you're like, what amino acid sequence makes this? And as you said, there could be multiple amino acid sequences that make that particular structure. And I think what this tool is doing is that at each point of the beads in on the string, maybe it's like predicting one amino acid at a time. And it's sort of going like, maybe let's try from the start of the string. And I think that's asparagine. And you're like, okay, that's that. And so given I know that, what could the next one be? And you're kind of building it up by thinking about the larger structure. But also now that you have already predicted some of the previous amino acids on that string, that gives you a bit more information about what the next amino acid could be.
Yeah.
And so you're using the information that you already have from the neighbors to predict the next one. But sometimes that probably leads to you like getting stuck in dead ends, because sometimes it doesn't work.
Right.
And so maybe that's why it produces many predictions, and then some of them won't be that accurate.
It could be mispredicting the structure. And what a lot of people who work on protein improvements like this want to do next is to run a check on, hold on, is this going to end up folding up like I thought it was? And I think you probably know how they do that.
Ooh, is it AlphaFold?
It's AlphaFold!
That makes sense. Okay, so just recapping. Protein MPNN is predicting the amino acid sequence from the structure.
Yes.
And I knew that AlphaFold predicts the structure from the sequence.
Yes.
And so if you have predicted the sequence and then you're like, wait, does this actually fold into that structure that I started out with?
Yep.
And so you would use AlphaFold then.
Exactly. You can kind of validate, does this fold up how I want it?
Right. But it's not truly validating, right? Because you would want that to be done in a lab.
Absolutely
probably.
Right. And so what I did in this toy project was just the steps we just described. And I did not have enough time to validate in the lab whether the tweaked protein molecule was what I thought it was, or was doing what I wanted it to. But what's amazing about these tools is that they've gotten so good that in combination, I was talking to PhD students and postdocs there who had sort of been in the lab before the deep learning revolution, been in the lab during and after the deep learning revolution. And they're like, everything's changed. Like now they can generate, say, their 50 favorite protein sequences that are hypotheses that they've validated with AlphaFold, but haven't truly validated. And they can say, okay, I'm going to order those up online and I'm going to validate them in the lab like next week or the week after.
You mentioned the change between now and the grad students
Yeah.
before. And that kind of reminded me of like what it used to be like to do this.
Oh,
And I was recently reading this book called The Code Breaker, and it's just about like, Jennifer Doudna and how she developed CRISPR and stuff like that. But the author actually starts kind of much earlier on, and he describes what's happening in the 1940s and 50s when people are discovering the structure of DNA. And this is relevant because I think when Watson and Crick were trying to figure out the 3D structure that led to DNA, they had to actually use these like physical models of like
yeah.
atoms in a lab. And so they'd have like these like balls and stick type figures, and then be like, okay, what angles are working, or like what is going here, what is going there, like what are the specific atoms that is making this DNA. And they didn't even know that it was a double helix at first. I think they thought it
Right.
was a triple helix.
Yeah.
And they had like one of them had just misremembered the amount of water that was in the total molecule or something, and that got them confused for a bit. But that's sort of what would be happening here before, where if you're trying to figure out the amino acid sequence of a structure initially, and I think this was maybe pre 2000s or something, people would be actually making physical models of the same and like, does this work if I put this amino
Right.
acid here and then the next one there, and like, does it fit together? Is one of them like negatively charged and the other one also negatively charged? Like,
Yeah.
how does it all work? And that sounds extremely complicated when you have a much bigger protein. Or I mean, it's just complicated, and then there are so many possibilities.
Yeah. So we no longer have to go physical ball by
Right.
physical ball.
Yeah. So I think there were some things between that and now. There were some kind of statistical modeling techniques in the 2000s and 2010s, and then there was some like AI tools and deep learning and stuff like that that was used as well. But this is really different because I think it's trained on data of the structures of proteins. And what that means is like over time, people would be working in the lab to figure out exactly what a protein looks like with like X-ray crystallography. So you're like crystallizing the protein.
Yeah.
You're getting an X-ray image. You're trying to determine what it actually looks like, or, various other techniques. And then they are trying to figure out again, like, what does this look like? And then because you would then figure out the coordinates of the different atoms, you then have a lot of data that's collected over the last few decades that has gone into this database called Protein Data Bank. And I think that's the biggest one.
Yeah.
And using all of that data of like the coordinates of the atoms and like which amino acid sequences there are, and then maybe like similar types of proteins and things like that, you could then make these predictions a lot better. And so I think that's what this is doing. It's this like graph-based deep neural network. And so it has data on like the coordinates, which you've also put into it with the overall structure that you have. And then it's like which amino acid sequence is in each part of the chain.
Yeah. And that data we're talking over 100,000, I think it was 170,000 structures that AlphaFold 2 trained on.
So that's individual structures that people have worked out in the lab through these other methods like X-ray crystallography and making these physical models.
Yeah.
Which is crazy.
That's a
huge amount.
It's a huge amount. Decades, probably 50 years of work of thousands, tens of thousands of grad students, postdocs, professors, mostly on public funding.
And I guess I'm imagining in the past, you might just be an individual person who's like, let me try to guess what the amino acid sequence is based on stuff that I've learned. And that's much harder than if you just are this computer model that's like trained on so much of it. You don't have to try to remember each of the structures.
I can do it, but I know a lot of other people struggle. Yeah. Yeah. They really did it different.
Okay, so quick recap. we have this protein in the Streptococcus bacteria that we want to make and we want to change a bit. What was the change that you mentioned that we're doing here?
We're going to make it smaller and just use a couple of its domains.
Okay. And okay, so we're only using a few domains, and that should be enough for us to recognize it without causing other problems for us.
That's the hope.
All right. And then what's next?
Well, what you would do next that I didn't get to do because I had to move on would be trying to validate what protein MPNN and AlphaFold have helped you get to as hypotheses, but validate them in the lab.
So we've got the domains that we want. We know the structure that we want. We then have figured out the amino acid sequence that leads to that. And then we want to check, does that sequence actually create that structure?
Yes. And the asterisk I would give, though, is that we have hypotheses of multiple sequences where they're a little bit different because protein MPN is giving us some hypotheses.
Right. And some of them might be wrong.
Some of them might be wrong. Some might be better than others. And a lot can change with just, one amino acid change here or there.
Right. As you said before, that one amino acid change means that it can no longer cut up or signaling proteins.
So what you would do here, and as a spoiler, after I'd done all this thinking and computational stuff, I was like, before I order these up, let me just check if someone has done this. And you'll be amazed to hear that a group at Shandong University in China had already done all these experiments like 3 years ago.
Aww.
It's terrible to have an idea.
So they had already come up with this slightly adjusted version of this protein with just those domains, and they had tested that it worked in a lab. And did they?
They had done the subdomain analysis. I'm not sure which alterations they'd made and if they used AI for those alterations, but the basic punchline of what they did was, let me walk through what validation would look like here. So at the very end of the day, the validation we care about is if you inject this or inhale it or something as a vaccine, will it protect you as a human being against a future Strep A infection? We're not going to test that. We're going to test some earlier things to get an initial idea. And so what you can do is you can order, let's say we had 50 hypothesis amino acid strings. You can order up the DNA that would code for each of those amino acid strings. So remember from our first episode, we go DNA to RNA to proteins. So we want, the DNA sequence.
So we've basically made each of the sequences that protein MPNN has spit out.
Yes.
We've made like a bunch of them.
We've made a bunch of them, say 50, and we're getting the DNA strings. You can order those online or, from your favorite provider, Twist Biosciences or, and IDT. You can get them delivered to the lab maybe next week. They'll be pretty quick. And you can then take those DNA strings and try and grow up the proteins by putting the DNA inside a living system. So say bacteria like E. coli or yeast. Or
what is why are you telling this protein to grow up?
I'm telling you to grow up, Salome. I am telling the protein to grow up because I need to use it in experiments.
But what is growing up? What is
growing up?
What is a baby protein?
A baby protein is, more like a butterfly situation here, where I want the butterfly, and so the caterpillar is a string of DNA, and so I'm gonna have to get a little cocoon going, and that cocoon in this case is a bunch of yeast cells that I've put in a little vat, and I'm going to sort of feed nutrients to, and I'm going to jostle around for 2 days, and they're going to have a good time in there.
That's really cute.
And by the end, I'm going to slice them up, and I'm going to take the protein.
It's got really violent.
I would never hurt a butterfly, but a yeast cell, watch out.
Okay, so you are trying to recreate this protein by growing it up in a bacteria from the DNA.
Yes, exactly.
And you have now made lots of them.
Now I've made lots of them, and here's what I might do with them. Experiment number 1, I would probably take a bunch of those proteins and inject them as if they're a vaccine into some mice.
Oh.
And the mice are going to have some sort of response to that, or maybe they won't. And the response I'll first check is, take a sample of blood from the mice, say a month later, and see, okay, did any antibodies get produced?
Mm-hmm.
And if so, sign number one, we're headed in the right direction. If not, this ain't so good.
Uh-oh. And, well, you're not just testing, does it produce any antibodies? You're like, does it produce antibodies against a specific protein?
Absolutely. So, the next experiment I would do is against the specific. What I would do is take a wild type of the protein. So don't take this thing I just grew up.
Mm-hmm.
Take, get some Strep A bacteria and take this, isolate somehow the...
Right. So does the mouse now create antibodies against the protein that I was trying to use as the target?
Exactly. So I would take the sera, take the blood and say, are the antibodies produced? Let's say there were some produced, so that's immunogenic.
Mm-hmm.
Are they cross-reactive to the wild type of the protein?
Right.
So do they bind it tightly? Do they bind it a lot?
Yeah. So did your little protein vaccine actually help the mouse protect itself with antibodies against the natural protein?
Yes, but I would go even one step further, which is, so that's next experiment, but to protect the mouse, you actually care about, is it protected against the bacteria, not just the protein. The third experiment's still in a dish. I'm still taking my sera that has antibodies in. I'm putting it against the bacteria. And I'm seeing, does it bind the bacteria or neutralize bacteria? The best thing after that would be, okay, we injected this mouse and now we're going to challenge it with some sort of...
Oh.
I know, with a bacteria.
Right.
And then some of them
are probably going to die.
Yeah. So
how dangerous is this? Is this infection?
For most humans, not super dangerous.
Mm-hmm.
So probably true for most mice.
Okay. So you've now checked that a different research group has made this and validated it, and now you know that protein that you improved could have worked.
Yeah, I mean, it's basically, it turns out that of the 5 domains that make up that protein, if you take some subsets, it's not looking good.
Mm-hmm.
And you take some other subsets, it's looking pretty good.
Okay.
And so it actually, looks like it's probably worth doing. And if you take those subsets that look like maybe they work, you can then create a vaccine involving maybe just that, or involving other, subsections of other proteins that might be antigens. And
That's really cool.
It's really cool. And
so, but this would still just be the start of the whole process of developing a new vaccine. You would then need to test it in human clinical trials, like phase 1, phase 2, phase 3. That could take another 8 years, 10 years, something like that.
And AI has not sped up that yet.
Right.
So there is more to do.
Has this been done before? Like, are there drugs and vaccines already that have been improved with
is
AI?
A drug which was made during the COVID pandemic by Neil King at the Institute for Protein Design and David Wiesler and students there, that used predecessor tools to create a vaccine using just the RBD region, receptor binding domain of the spike protein.
Mm-hmm.
So the vaccines that I got, what did I get? I got Pfizer followed by Moderna maybe.
Mm-hmm.
And those are using the full spike protein.
Oh.
And it was pretty good. But it also is, if you can get away with not using the full one, you might be able to do even better.
Right.
And sure enough, Neil King managed to say, I'm going to use the really important domain, the receptor binding domain only. And I'm going to encode it on a nanoparticle, but like a soccer ball. And I'm going to stick out a lot of them. And it's going to generate a really good antibody response.
That's very cool.
That happened within the last 5 years.
Wow.
It's wild. Yeah.
I'm also thinking, it's kind of reminded me of the RSV vaccines. Were they also improved with AI, I think?
The story I remember there is Jason McClellan at the VRC in 2013. Mm-hmm. So my guess is that main breakthrough for RSV predated the deep learning revolution. But I don't know the story of, it probably did involve cryo-electron microscopy, which probably was helped by ML.
So I think what I remember is RSV, which is respiratory syncytial virus. It's a lung infection. That's one of the most common reasons that infants go to hospital in the US. In the 1960s and 70s, people tried to make RSV vaccines. And there was this protein on the surface of the virus that they, used as a target. But it wasn't working very well. It had a lot of side effects. And in some cases, it was actually making the infection much, much worse when people got infected. And so a lot of people just gave up at that point. And it was just seen as this is this unsolvable challenge. We're not going to be able to develop vaccines against RSV. Sorry.
Mm.
And then something changed in the 2010s, which was that this type of electron microscopy became much better. And I think it was the software improvements that you could then figure out at a much higher resolution what these proteins looked like. And so what they figured out was at the surface of the virus, the protein looks a certain way before it infects the cells, but a different way after it infects the cells because it uses that protein to fuse to the cell and enter
Yeah.
it. And unfortunately, the previous vaccines were using the after version
The
of
post.
the protein.
Yeah.
But that doesn't work because if the virus is like swimming
late.
around in
Yeah.
your blood and your immune cells only know what it looks like after it's entered, well, that's too late. You
Yeah.
need to figure out what it looks like before. And so they figured out what it looked like before it entered the cell. And that is called like the pre-fusion version of the protein. And in order to remake vaccines with that version, I think they used AI to introduce stabilizing mutations and keep it that way.
Right.
And now we have at least 3 RSV vaccines that have been approved in the last 2 or 3 years. This breakthrough in the 2010s with microscopy, man, suddenly we know how to design an RSV vaccine now.
Yeah.
And so
0
there were
to 1.
multiple people who were like, oh, well, now we can do it. And so it wasn't just it wasn't just one person who made the breakthrough, but I think this was the big breakthrough with the microscopy.
And that big breakthrough.
And AI.
And AI. The microscopy breakthrough happened on public funding at the NIH Vaccine Research Center.
Whoa.
And then the vaccines there are now many vaccines.
Saving a lot of babies.
And saving a lot of babies. And so that's my pitch, implicit pitch, that often you need a breakthrough and often it takes researchers who aren't trying to go after a product, they're actually trying to understand something.
Yeah, and something might look like an unsolvable challenge for decades, and then something changes, and now 3 people can do it.
Yep. So, stepping back to summarize the whole story here. We started with an invader we want to make a vaccine against. We took a protein that might be an antigen, and we started tweaking that protein with the help of a couple AI systems, Protein MPNN and AlphaFold, to come up with some hypotheses of subunits of that protein and smaller, versions of it that could be vaccines.
Mm-hmm.
And the initial results are that maybe some of them actually look promising and should be taken further and explored further.
Right. And so where is the one that you talked about, the CypA? Where is that in clinical trials right now? What's going on?
Not yet, because everything is slower than it should be in vaccine design, especially for global health. But I would say it's one of probably the top 10 antigens that people are exploring pretty seriously.
Mm-hmm.
And people are looking at combinations of those antigens in 4 or in 5 different proteins in combination with other adjuvants that help prompt an even stronger immune response. And I'm cautiously hopeful that one of those combinations will actually prove to work.
Just to indulge you a bit on Strep A, what is the reason that, what's going on with the field? Why don't we have much more interest in this? And how far away are we from a vaccine?
I think that there are a couple answers to why there's no more interest. The really predominant one is that the most of the deaths that strep A leads to, so the biggest forms of harm, occur in countries that are not wealthy. So there's not as much of an incentive as there could be for pharmaceutical companies to prioritize it. That said, there's a pretty decent incentive because a lot of parents with young kids are not exactly fans of strep and would be probably quite excited if there were an available vaccine just to prevent strep throat or sore throat, pharyngitis, and scarlet fever and all of that. I think there are other reasons too. And what you really are making me want to do is a whole episode on strep because it's so interesting. To answer the last part of your question, I am actually fairly hopeful that this should be a solvable issue. I think you should be able, with modern techniques, to design around some of previous problems, and you should be able to test whether these things work and get an answer. And what we care about most is figuring out how they work in children so that you can prevent these repeated infections that lead to problems.
Right. And so we talked about AI being used to improve or like cut up this protein into a specific domain and then see if this domain is still soluble, and if not, like improve that solubility. And then I also mentioned RSV vaccines, where AI has been used to stabilize a particular protein that's used in the vaccine. What other uses of AI is there when you're applying it to proteins and improving them?
Two more come to mind of properties that are often really nice for vaccines. So one is thermostability. Human proteins have evolved to behave very well at human body temperature. If you're shipping a vaccine around the world, sometimes you're in colder and sometimes you're in hotter temperatures than the human body. And so if you can make sure that won't denature your protein, then you're going to still have a useful vaccine out the other end. So you might want to make some tweaks for that. And then another one is, we talked about immunogenicity a bit earlier of, does this prompt any antibodies, for example? You also care about what's called immunofocusing sometimes, where can you present the parts of a given protein, say the epitopes that are most reactive, as much as possible or in the right geometry, so that you can target the response to the most productive bits. So it's not just you prompting any antibodies, it's are you prompting the right ones, are you prompting it as frequently as possible and binding as tightly as possible.
Right. So these are all ways of making sure that our immune system recognizes the protein or the whole pathogen well, how well it's doing that, and then also maybe kind of optimizing the way that it's doing. Like, I think you mentioned this at some point when we were talking earlier about how sometimes if you are recognizing a pathogen, that might confuse your immune system because parts of a pathogen might look like other parts of your body. And then you could develop an autoimmune reaction to other parts of your body because your immune cells have treated that as like foreign as well from after seeing the pathogen.
Yep.
So we're trying to do all of these things potentially.
All of these things.
Is there anything else? Are there other uses that we would have for AI?
I bet you there are, and I can't wait for listeners to write in and tell us or start working in their basement on some of these applications.
Cool. Right. But this is also only the beginning. There has to be a lot of testing and stuff before things get to the market that can be so that it can be used for people. And then also there's lots of stuff that happens before this process, like collecting all of the data in the first place, doing the laboratory research, figuring out these structures, making sure that there is data for AI models to train on and help us make these improvements.
Okay, so we're improving proteins found in nature. What about if we could design entirely new ones never seen before?
I want to do that.